Hello everyone. In this chapter of Life on Planet Earth, we are going to address the cell. Specifically, we are going to address the structures and the functions of the different components of the cell. We are going to start this chapter with addressing the cell theory. What is the cell theory? The cell theory is the idea widely accepted by all biologists that all living organisms are made up of cells. That's the first idea. The second idea is that regardless of the organism, the cell is the basic unit of life, meaning that the cell is the smallest component in a living system that carries all the characteristics of life, including reproduction, growth, response to environmental stimuli, adaptation, evolution, use of energy, and the necessity to acquire nutrients, to mention only these characteristics. In addition to the cell being the basic unit of life and that all living organisms are made up of cells, you have the third segment of the cell theory, which is that all cells come from pre-existing cell, which always raises the question of the origin of the first cell. But let me close that parenthesis. As we consider the cells in a multicellular organism such as a human, we are going to have different kinds of cells. We have muscle cells, we have nerve cells, liver cells, skin cell, you name it. All these different cells are very specialized cells. This specialized cell, because they play a very specific function in the body, they are said to be differentiated cells, as opposition to stem cells. Stem cells are cells in our body that are blank. These cells that are blank can be differentiated into specifically a skin cell, a nerve cell, a liver cell, a muscle cell, right? If we consider, for instance, the stem cells that are found in the red bone marrow, these stem cells are already predetermined to become blood cells, whether you are talking about red blood cells or white blood cells. And there are different kinds of white blood cells, right? But I'm not going to go into the details of that. What I want you to remember, however, it's that all cells in our system that are differentiated started as stem cells, right? Now, you have different kinds of stem cells. I'm not going to take you into the details of that. With that being said, when now looking into all the cells that we can encounter across all the living systems, these cells fall roughly into two categories. We have the prokaryotic cells and the eukaryotic cells. The prokaryotic cells in general are going to be small, simple in terms of organization, right? And their genetic information typically is not enclosed in an envelope. Because of that, the genetic information is sitting in a relatively open space in the cell. That open space in the cell is called the nucleoid, where the genetic information, DNA, is found. And this is the case for bacteria and another type of prokaryotic organism that are archaea. Then we have eukaryotes. Eukaryotic cells are the cells that are very complex. Complex in the sense that they are going to have not only their DNA, the genetic information, enclosed in an envelope. The whole structure is called the nucleus. In addition, you are going to have many sacs inside of within the limit of the plasma membrane, which sets the limit between the inside and the outside of the cell, we're going to have many organelles, specifically membrane-bound organelles, right? Because of that complexity, the many compartments in eukaryotic cells that cause eukaryotic cell to stand out compared to prokaryotic cells. So we are going to go into more details when we look at the different types of cells. Okay? So the question I would like you to ask as we are going through this chapter is, what are the different components that make up a cell? And what are the functions of these different components, right? Also, and as important, it's what are the basic structures that are found in a living cell, whether you are talking about prokaryotic cells or eukaryotic cells. What are the basic components of a cell? These are things we are going to address in this chapter. Before we address this thing though, know that cells are very small. The cells in our system, they are very small. On average, a cell in a human is going to be about 50 micrometer in terms of diameter. Now, the smallest particle that you can see with your naked eye, it's about 0.2 millimeter, right? Now, that 0.2 millimeter, it's also equal to 200 micrometer. Now, 200 micrometer, which is the smallest particle you can see with your naked eye, is much larger than 50 micrometer. Now, some cells are way smaller than 50 micrometer. Some cells are slightly bigger, but you hardly have any cell that approaches the 200 micrometer. Cells are very small. Because they are small, we need tools to observe them. These tools, the most common, actually occur in two kinds. One is the light compound microscope, and the second type of microscope, it's going to be electron microscope, right? Here we have a scientist actually using an electron microscope. There are two kinds of electron microscope. We have the transmission electron microscope, transmission electron microscope, and we have the scanning electron microscope. The micrograph that you see here on the upper left corner, it was taken with a transmission electron microscope. A micrograph is a photograph that is taken with a microscope, right? Now, the red line that you see here, it's the limit of this eukaryotic animal cell that is observed under an electron microscope. Now, the transmission electron microscope has this characteristic that it can allow us to see the fine details inside of the cell, right? So that we can see, obviously, the plasma membrane in red. We're going to see right here the nucleus. And inside of the nucleus, we have some DNA, but I'm not going to bug you with that. We have a number of organelles that appear green here. These are little sacs containing substances, right? The gray part here is the cytoplasm. All right? So The transmission electron microscope allows us to see fine details inside of cells, and also these fine details appear in what is a two-dimension image, 2D image. Now, how about a scanning microscope? A scanning electron microscope, SEM, allow us to see the fine details on the surface of a cell. Now, here we have the a micrograph that represents the scanning electron microscope image of a stem cell. We can see the fine details of the surface of this stem cell. Now, more common, the light compound microscope. Light compound microscopes are the most common microscope that you are going to see. With the light compound microscope, you can see organisms, cells. However, you are not going to see them with the level of details that you would see with an electron microscope. And by the way, an electron microscope uses an electron beam to be able to observe cells, objects, while a light microscope uses a light source to be able to observe organisms or cells. So here you have a euglena. Euglena, it's a type of protist. It's a single-celled organism. Eukaryotes can be made up of unicellular organisms, or they can be made up of multicellular organisms, such as a human. But euglena, it's a unicellular organism. All right. So this slide here gives you an appreciation of the relative size of objects, right? The smallest thing we can see with our naked eye, I was telling you earlier, it's 0.2 millimeters, right? Which is 200 micrometers, right? Now, notice that a chloroplast, it's 1 micrometer, while a plant cell and an animal cell, they have a size that fall between 10 micrometer and 100 micrometer. So these cells are very small, but we can see them with a light compound microscope, right? Anything that is smaller than the chloroplast, you cannot see it with a light compound microscope. You have to use an electron microscope, right? So that you cannot see viruses with a light microscope. You need an electron microscope to see viruses, to see large proteins, right? And even amino acids, actually, you can see with an electron microscope. Now, going back on the spectrum that you can see, you're going to have a frog egg. You can see a frog egg. You can see an ant, right? That is about 1 centimeter. That's the size. While a frog egg would be about 1 millimeter. A mouse is going to be a matter of multiple centimeters. A human is going to be taller than a meter, right? 1 meter. And a blue whale, it's multiple meters, right? Now, with that being said, what is the size of the largest cell? Now, I'm not going to tell you exactly what the size of the largest cell is because I don't know the size of the largest cell. However, what I want to attract your attention upon when I ask what is the largest cell, it's that some organisms actually have humongous cells, right? Such organisms are the giant squid right here and the colossal squid right here. This squid actually can weigh up to a ton, if not more. Now, think of the nerve cells in this organism, this squid, right? These nerve cells that expand from, say, this part of the body to the very extremity of the body here, that cell has to be huge, right? Now, while you have some cells that can be large, including the cells in the giant squid and the colossal squid, most cells are small. When you compare, for instance, the cells in a human and the cells in an elephant and the cells in a mouse, in terms of size, they don't vary tremendously. One can wonder, well, why then the elephant is so big compared to a mouse? Why is a human so big compared to a mouse? Well, it's boiled down to the number of cells. It boils down to the number of cells. Now, I'm not saying that some of our cells, including nerve cells, right? For instance, a nerve cell that expand, say, from the shoulder, right, to the length of your arm, right? This nerve cell may be somewhat long, right? However, seeing them, it's another story, right? The question we may ask is, why are most cells small in size? Most cells are small in size because the smaller a cell is, the higher the chance of interaction between chemicals that are found in a given space, right? So, for instance, if you consider this cube here, it's one single cube for and each side it's 4 centimeter. So that's 1 4 centimeter cube, right? That 4 centimeter cube has a certain surface area to volume ratio, right? However, when we split that one single cube into small cubes, so 8 cubes of 2 centimeters, what happens is that while the volume remains exactly the same, the surface changes. The surface increases because now you don't just simply have this surface, right? You also have this surface that is hidden here. You have this surface for this cube. You have all the surface for this cube. You have all the surface for this cube. And you have all the surface for this cube, and so on. So you are increasing the surface while you are keeping the same volume. Now, if we split the cube even in smaller size, now we are going into 64 cubes that each has 1 centimeter. We are even increasing more the surface so that the surface area to volume ratio increases from this cube right here to this cube, 1 2 3, to this cube right here, where the surface is much higher than you do have here, right? This has the advantage that you are going to have the chance for two chemicals to meet each other more easily here, right? Than they do have the chance to meet here for the same number of chemicals, okay? So the next step is going to be looking into cell structures. And the first structure we are going to start with is the plasma membrane. Now, what is the plasma membrane? The plasma membrane is that structure in the cell that sets the limit between the inside and the outside of the cell. this plasma membrane is very complex and it has a certain number of properties that we are going to address. Now, the different molecules that make up the plasma membranes are going to be the phospholipids. The phospholipids are going to be organized in such a way that they are going to form a bilayer, right? So we have one layer of phospholipid here, and we have another layer of phospholipid here. That's a phospholipid bilayer, right? Now, what I want to attract your attention upon here, it's the characteristics of phospholipids in terms of physical behavior. In terms of physical behavior, the head in red here is hydrophilic, right? It's a hydrophilic head, so it can tolerate water. The tails here are hydrophobic. They do not tolerate water. Now, when phospholipids are present in large amount in a water-based environment, spontaneously, they are going to form structures that look like this, right? Setting the inside apart from the outside. All right? So that's exactly what the plasma membrane does, right? The phospholipid bilayer sets the limit between the inside and the outside of a cell. That's one. Two, we are going to have the proteins that are embedded. These proteins that are embedded play multiple roles. We are going to address these roles at a later time. In addition to the proteins that are embedded in the plasma membrane, we are going to have proteins that are embedded and some sugars are attached to these proteins, right? Carbohydrate chain. Now, together, the carbohydrate chain and the protein gives the name of glycoproteins to these proteins. Glyco means sugar, proteins for protein. So these are proteins that are attached with sugars, okay? So then, in addition to these glycoproteins and these proteins, we are going to have cholesterol, okay? Cholesterol is going to play a very important role at modulating the fluidity in the plasma membrane. I mentioned that word, fluidity. I want you to remember that the plasma membrane is fluid. Because the plasma membrane is fluid, the chemicals can move in and out. In fact, the plasma membrane is semi-permeable. Semi-permeable because it allows some chemicals to go in and out and vice versa, while not allowing some other chemicals to go in and out freely, right? Nevertheless, the cell has to acquire some material from outside and has to send some material outside. How does the cell do this? That's another question, right? So I want you to know the components of the plasma membrane, including the phospholipids, right? Know that the phospholipid make up a phospholipid bilayer with a hydrophilic head and hydrophobic tail. Now, if you were to cut the plasma membrane with a knife, okay? If you were to cut the plasma membrane with a knife like this, right? What happens is that you are going to hit first hydrophilic heads, then hydrophobic tail, then another hydrophobic tail, then hydrophilic heads. I want you to remember that, okay? That organization of the plasma membrane is very important. I also want you to know that the lipids actually move in the membrane. They have movement that go from side to side, right? They have movement that go like this, okay? They also have movement of rotation. Movement of rotation are like this, right? So they kind of go around, okay? And they have another type of movement that is called flip-flop. So they can go from the top side to the bottom side, and from the bottom side to the top side. They can actually do that. But I'm not going to bug you so much with that flip-flop movement. What I want you to remember is that the plasma membrane is fluid. That's one. Two, it looks like a mosaic. So the plasma membrane, it's a fluid mosaic model. A mosaic, basically like an artwork, right? A mosaic is made up of many small pieces, right, that are brought together. When you look at it from very far, the artwork looks great. But when you come closer, this is when you realize that, oh, what I see from far as, one single nice piece of work, it's actually many small pieces connected together when you get close, right? This is what you see when you look at this protein here in the ocean of all these phospholipids, right? This looks like a mosaic. So it's a mosaic, a fluid mosaic, right? Fluid, but look like a mosaic. All right. So let's address the proteins that are embedded in the plasma membrane and their role. Now, the proteins that are embedded in the plasma membrane are going to play many roles. Some of them are going to play the role of channels, right? Some are ion channels, allowing chemicals to go inside of the cell and outside of the cell. Without these channels, whatever chemical is going through that channel could not go through the channel. In fact, a disease such as cystic fibrosis, right, that causes thick mucus in the airways, right, is caused by a defective channel. That defective channel is supposed to carry chloride ions across the plasma membrane. But let me close that parenthesis. Just know that a defective channel can cause health issues, including cystic fibrosis. I want you to remember that. Now, another function of proteins that are embedded in the plasma membrane, it's transport. So we have transport proteins. The transport proteins are going to carry chemicals from the inside toward the outside, or from the outside toward the inside, right? Either way, these proteins are very selective, including also the channel proteins. They are also very selective as to which chemical they are allowing in and which one they are allowing out. So remember that the channel, the transport proteins, they are very selective as to what chemical they are carrying. Then we have the cell recognition proteins. These cell recognition proteins allow, for instance, our cells to recognize that I am in the right context. How does a white blood cell know that this is another white blood cell over there, right? And that this is a red blood cell over there, and that this is a liver cell over there? Well, they know that because you have on the surface of each of the cells some proteins that are modified with a sugar residue that is very characteristic of the cell. A liver cell is going to be different from a muscle cell based on the surface markers. That's not the only difference though, right? Some proteins are going to play the role of receptor proteins. These receptor proteins are going to attach a signaling molecule that comes from the outside, for instance, a growth factor that comes and wants our cells to multiply. That growth factor is going to be released and travel and come and attach here in an area that is pre-designed to match the signaling molecule, so that the cell can now receive the signal, send the signal inside, and a response is going to be developed. Okay? Then we have some membrane proteins that play the role of enzymes. These enzymatic proteins are able to change a substrate from the substrate to a given product, just like you see here, from this to this. Okay? And then we have junction proteins. Junction proteins are these proteins that allow two neighboring cells to attach to each other and stay attached, to remain attached, right? For instance, the skin cells are attached to each other so that your skin is waterproof, right? But how are these skin cells attached to each other? They have junction proteins that bring them together, right? This is the plasma membrane of one cell, and this is the plasma membrane of another cell, okay? And they are connected together through here, all right? So these are the functions that are part of the membrane proteins. Now, the plasma membrane sets the limit of the cell. It controls the movement of chemicals in and out, right? It connects cell to another cell. It senses what is going on inside and outside, right? Senses what is going on inside and outside. It does provide enzymatic activity.